Dr. Lee.
And I'm Dr. Wang.
And I'm Dr. Kim. And our topic is the development and use of monoclonal antibodies against tumor antigens in the treatment of several types of cancer.
That treatments usually fall into one of the following categories: surgery, radiation, chemotherapy, immunotherapy, hormone therapy, or gene therapy. Monoclonal antibody is a part of immunotherapy.
Dr. Kim, what is monoclonal antibodies?
Good question. Dr. Lee, why don't you come over and answer this question?
Of course. Monoclonal antibodies, which are also known as MABs, are used in biological therapy for cancer treatment. They are also designed to recognize specific proteins on cancer cells. Each mononuclear antibody recognizes one particular protein. The attachment of a mononuclear antibody to its antigen triggers the cell to destroy itself. This in turn signals the body's immune system to attack and kill cancer cells.
Monoclonal antibodies are highly specific. Certain antibodies will only bind to a specific epitope. For an example here, antibody A will only bind to epitope A. Antibody B will only bind to epitope B, and so on with antibody C.
Dr. Kim, how are monoclonal antibodies made?
Very good question. The first step in preparing monoclonal antibody is to inject a mouse with an antigen that has a specific epitope of interest. The antibodies in the mouse are derived from a single cloned B cell, and each B cell produces a single type of antibody. B cells are then isolated from the spleen of the mouse then fused with myeloma cells. Myeloma cells are a type of cancer cell that grows continuously. They will then fuse together to form hybridomas. Myeloma cells, B cells, and hybridomas are then cultured in one plate under conditions that will only permit growth of hybridomas. Each of the hybridoma cells will produce a single type of antibody against a single epitope. Each type of myeloma cells are then removed into individual wells on the microtiter plate, and then tested for ability to produce the desired antibody. The hybridomas that's producing the desired antibody are then cultured, isolated, and purified. Now let's move to the lab and see how monoclonal antibodies are actually made. Before starting the procedure, you have to make sure that your hair is pulled back. Put on goggles, have the lab coat, and most importantly, put on gloves. Also, wear closed-toe shoes.
Tumor antigens are produced in tumor cells. It triggers an immune response. Tumor antigens are useful in identifying tumor cells and are potential candidates for use in cancer therapy. Among tumor antigens, some may be tumor-specific, while others may be expressed by normal tissues. There are 5 types of tumor antigens that we will be discussing. The first type of tumor antigens are known as undefined antigens. They are found in allogeneic and autologous vaccine. Examples of this type of vaccine are based on undefined antigen on intact cells, on cell lysate, on the total amplified RNA vaccines, and on heat shock proteins. The second type of tumor antigens are known as defined antigens. They have the ability to correlate specific immune responses with the antigen used, thus improving immunogenicity of vaccines. The vaccine will have to be correlated according to human leukocyte antigen types. Now let's take a look at a chart that describes advantages and disadvantages of undefined and defined antigens. Undefined antigens have availability of several potential tumor rejection antigens. Also, they have unrestricted HLA patient population. Advantages for defined antigens include temporal monitoring of specific immune responses and possibility of correlation of immune response with antigen expression on tumors. Now let's take a look at disadvantages. Disadvantages for undefined antigens: they are difficult in correlating clinical responses and overall immune response based on select known antigens. Also, they are largely dependent on clinical endpoint. The 2 disadvantages for defined antigens are limited number of known tumor antigens for use and relatively limited targeting of patient population due to HLA restriction.
Unique antigens are mutations in genes that could create new products. Even if there's a single mutation that occurs, it could increase the signal transduction, which leads to uncontrolled cell division.
The 4th type of tumor antigens is known as differentiation antigen. These antigens are molecules which are present on tumor cells which are highly specific for immunotherapy. Another way to describe differentiation antigens is a cell surface antigen determinant which is found only on cells and at a particular developmental stage. The 5th type of tumor antigens are known as overexpressed antigens. These are antigens on cancer cells that form certain attractive targets. So overall, these tumor antigens are identified by T cells and provide for the development of immunotherapy of various malignancies. Now, let's travel through the body and find where these cancer cells are hiding.
Hello, I'm a cancer cell. Would you like to see how I can be destroyed? Well, great, that makes one of us. Here's the first way monoclonal antibodies can destroy me.
Monoclonal antibodies are injected and then seek out cancer cell proteins. These monoclonal antibodies then bind to the proteins found on the cancer cells. The antibodies then signal the immune cells found in the cancer cells. The immune cells arrive and punch holes into the cancer cells, and thus leading the cancer cells to die.
Oh, hello. I'm filling in for my buddy. He, didn't make it. But I'm here to tell you the second way monoclonal antibodies can destroy us.
Cancer cells normally grow and divide with the use of proteins which bind to the receptors. These proteins then let off a signal to tell the cancer cell to divide. However, monoclonal antibodies block the signal by attaching themselves to the receptors Since these monoclonal antibodies bind, no signal is sent to the cancer cell to divide, thus leading the cancer cell to die.
Oh my gosh, this monoclonal antibody will destroy our entire population! And I'm here to have a good way to finally get rid of us. Oh no! Run for your life!
Radio-labeled monoclonal antibodies seek out cancer cell proteins. These antibodies then attach to the proteins, sending a radiotherapy which is delivered to the cancer cell, thus leading the cancer cell to die. As seen previously, cancer cells are destroyed by monoclonal antibodies. However, there are some side effects. A person who is injected with monoclonal antibodies can experience the following symptoms: chills, fever, itchy rash, feeling sick, breathlessness, wheezing, headaches, flushes, faintness, and of course, changes in blood pressure. Because these symptoms can occur, paracetamol and an antihistamine drug are given prior to an injection of monoclonal antibodies, which can reduce the outcomes of these symptoms. I hope you guys learned how monoclonal antibodies are used for immunotherapy and cancer treatment. Thank you for watching this video. Bye!